welcome everybody, and thanks for coming out tonight. This is our second Science on Tap event of the spring 2019 semester. My name is Jason Keller. I'm the interim dean of Schmid College of Science and Technology, taking over. All right. Yeah, technology and science both. Taking over the moderating duties from Andrew Lyon. And for those of you that knew Andrew, who ran these for the first couple of years we did them, he's now the founding dean of the Fowler School of Engineering on campus. It's really fun to do this. I was just talking to Maduka, and of all the things that I get asked to do as a dean, coming out and having a free beer and talking to people like Maduka is at the top of the list of things that I'm enjoying. So thank you for being here tonight. I want to start with our thanks. Thanks for Chapman Crafton for hosting us tonight. Thanks for Sarah and Elisa and our parent volunteers for all of your work in making this happen. Thanks especially to Bonnie, who apparently brought us all chips this evening. So I don't want to say we have a favorite parent volunteer, but Bonnie is right up towards the top of the list. And thanks to all of you for coming out tonight, mostly because you're keeping this place warm enough that I don't have to wear gloves. So I appreciate it. For those of you that haven't been to one of these before, this is a play in 2 acts. In Act 1, Maduka and I will talk a little bit. He'll share about his research, what he does, why he's excited about what he's working on at Chapman. There will then be a brief intermission where we will ask you to go refill your beers. And you'll notice on your tables in front of you there are note cards and pens. Once you get your beer refilled, we would like you to write questions on those note cards. Because during Act 2, we will sort through those note cards. Maduka will select a couple of those questions that he's excited about answering, and we'll spend the second part of this evening answering those questions. As an incentive to fill those cards out, if your question is selected, you will get a drink ticket, which you can use to get a free beer. So, you know, there's worse things. Okay. So it's really my pleasure to have to introduce Maduka Agba tonight. Maduka is an organic computational chemist, which, if you're paying attention, means he's exactly who you do not want to have a beer with, right? Like, if you are a moderately intelligent individual, you just said, oh my God, this is not who I want to have a beer with. And 98% of the time, you would be exactly right. But he is the other 2% of the time. It's going to be a really great conversation tonight. I think one of the things he does is take incredibly complicated computational and organic chemistry topics and make them actually really interesting and really accessible. So I'm looking forward to our conversation. So with that, thank you for being here. And I'll start with an easy question for you. Tell us a little bit about yourself. What's your background, and what brought you to Chapman University?
That's not an easy question. First of all, I just want to say thank you to all who organized, Jason, Andrew, for me being here. It's been a pleasure. So I got into Chapman last July, and I came here from Pomona College as a postdoctoral fellow. Before that, I did my graduate work in Oregon State, and beavers over there. And then before that, I did my undergraduate in Trinity in Texas, and then before that, I was born and raised in Nigeria. So I've kind of traveled quite the around the globe, I guess. So my field is in computational organic chemistry. So when I got into college, I knew I wanted to marry technology and medicine together. But I was one of those, just like any other freshman, I was very enthusiastic, but maybe a little bit naive about what the realities of life was. But I went to the chair of the computer science department at Trinity University, and then he had mentioned these 4 career paths. And he mentioned bioinformatics, cheminformatics, computational biology, and computational chemistry. And this was right at the first week orientation fair. And it was from there that basically, I think, set the direction of my life. Because he told me these 4 words, and I went to do my own research, and lo and behold, I'm basically here. So what do I do? So if you're thinking about a chemist, you usually think about someone who has a lab coat, wears a goggles, mixes chemicals in a vial, and maybe blows things up once in a while. I actually do none of that. And so the chemist that you're thinking about usually would have a molecule in 2 different vials, so 2 different flasks. And they will put the molecules together, and through some level of transformation, maybe they'll heat it up, maybe they'll mix it, and somehow some magic happens in between that first process, and then a whole new molecule has been made. My work deals with that magic. So what is going on between when you mix two molecules together and you get a whole new molecule again? And really the only way that you can really probe that, and there are many ways experimentally, but the way you can directly probe that is through theory and through theoretical chemistry. So I model chemical reactions. I try to figure out what happens during the transformation. And if I can do so, the idea is that we can then propose models for making that reaction go better or go faster, be more optimized. And so my entire field is basically dealing with that.
Awesome. All right. So you I'm going to read this. You gave us the title of tonight's conversation is Designing New Molecules, Lessons from Nature, which you're not fooling anybody. Can you help us unpack that a little bit? What are the sort of lessons that you're looking for? In talking to you before this tonight, this really frames the way that you think about your problems. And so I'd like you to tell us a little bit more about what you mean by lessons from nature and how you're really probing life to do what you do.
Yeah, no, thank you for that question. So the way I like to always think about or frame this is, so in the human body, you can maybe think about the human body as a factory. And it's usually a good analogy for me. And within that factory, you have thousands of these, I call them workhorses. And essentially, these are highly specialized, highly skilled workers that have evolved over millions and millions of years to do one specific task in the best possible way with the minimum amount of mistakes possible. Because one mistake can completely destroy the human body. And so the evolution of this has always fascinated me. What are these workhorses? How do they behave? Why do they do what they do so well with the level of intricacy? And so to put some words into these, those workhorses are called enzymes. And these are large, in my frame of view, molecules that essentially catalyze or make reactions go faster, or they actually just make reactions happen in your body. So I'm interested in understanding that process. What are the physical and chemical factors that govern these workhorses? Why do they work the way they do? And the goal ultimately is to take what we learn from nature, take what we learn from these enzymes, and design synthetic versions of them. So versions that are not in the body, not in living organisms, but emulate or mimic the efficiency of what those workhorses do in our body. So that's when I say we're learning from nature, it's a maybe not so modest proclamation that nature does what it does best, and it does it better than how we've ever imagined it. So I think the best way to do it is just try to emulate nature.
Awesome. And so my follow-on question here will be, can you give us some specific examples of the enzymes that you work with or projects that you're currently working on or excited to work on to sort of help flush out how this all works?
Yeah, no, so, well, first of all, I want to shout out, I just got to Chapman, and I already have two amazing students, Matthew and Lindsay, who are right there. Shout out, they're supporting my team. And both of them are working on fascinating topics. And so our research has sort of revolved around sulfur. So sulfur is one of the elements on the periodic table for those who look at those tables all the time. Me and Allegra over there. And it's the 15th most abundant element on our Earth's crust. So it's interesting, when I actually started thinking about sulfur was in graduate school, I was in a class project with a friend of mine, and we both collaborated for the very first time on delivering a particular project to a faculty member. He actually remembered that moment, and a year later, he called me up and asked me for advice on doing some theoretical work on an enzyme he was working on, which happened to have the element sulfur in it. And so we worked on that, and it was very successful. And then somehow our research started to merge. And so last year, I actually got another call for him. And so he's actually working at University of Oregon. And he's working on this bacteria called Helicobacter pylori. Don't worry about that. It's a pathogen that is found in the human stomach. And it's actually known to cause gastritis and stomach ulcers. About 50% of the world's population actually has this particular bacteria in their stomach. And what the stomach does, or what the body does, when it notices that this bacteria is in your stomach, it would start to pump out, we call these reactive oxygen species, but essentially species that would try to kill this particular bacteria. Most bacteria die based off of that. So that's how usually your body tends to cleanse itself from foreign agents. This particular bacteria has actually found a way to bypass this particular inflammation, so this particular reaction that your body does. And not only does it bypass it, it uses that signal to actually run towards that inflamed site in your stomach and suck out all the iron from your blood. So this is a very, very, really powerful bacteria. And so we call these bacteria kind of highly resistant bacteria, so bacteria that tend to be resistant to your body's natural responses. It's also resistant to the known drugs and known treatments. And so what Arden, this is my colleague at University of Oregon, is doing, is trying to figure out how does this bacteria figure out that there's an inflammation, sense this molecule, and then swim towards that inflamed site instead of swimming away. And then what is that mechanism in which it figures this out? Well, they found that, so this is now getting a little bit nitty-gritty, so excuse me for that, but inside this bacteria, there's this protein, we're just gonna call this chemoreceptor. Don't worry about that word. But this protein has a particular part of the molecule where you have a zinc metal and 4 amino acid residues. This is conserved across every single of similar protein in multiple different organisms. In this particular moiety, there's a zinc and a sulfur residue. This sulfur residue, in the presence of this inflamed molecule, so what the body actually kind of excretes from its stomach, will what we call oxidize, let's just say it changes form, and through that oxidation, that part of the bacteria actually flaps out, and that's how the bacteria senses that the inflammation is happening, and then it swims towards that inflamed site. Okay, so interesting problem. Once again, experimentalists, they would figure this out, and they say, wow, this is amazing. We figured out that this particular part of the molecule reacts to this inflammation in a certain way. Now, he came to me and asked, why or how does this reaction work? What is the mechanism in which this bacteria, that protein in the bacteria, modifies in order to react to and respond to the body's inflammation? So what I'm working on is actually looking at that particular portion of the enzyme, figuring out the mechanism in which that reaction works. And the goal is that if we know a little bit more about this response system, we can actually figure out much better, much more rational drugs or much rational treatments, and to try to shut down that particular process. But that's great for the biochemist. For the chemist, from my perspective, it's adding to my body of interest thinking about sulfur and how sulfur behaves in different conditions. And so it's a really interesting marriage between both of our research labs.
Awesome. That came dangerously close to being
know.
computational organic chemistry. So you pulled it out at the end there, and there was it was close. So I'd like to hear a little bit more about how you actually wrestle with a problem like that. So I am an experimentalist. I understand the sorts of experiments that you could do to test this and look for sort of chemotaxis. How do you do that on a computer? Because you're not at all an experimentalist. You don't have a lab. I don't even know if you have a lab coat. How do you do this on a computer? What does that actually look like for you?
So, getting too excited. 30 years ago, you wouldn't be able to take 2 atoms, or let's say 3 atoms in wherever, and compute that molecule. If a molecule is made out of 3 atoms, you wouldn't be able to do that with the hardware we have. But over time, because of the increasingly speedy and powerful hardware that we have, we can now compute molecules that have upwards of 300, 400, sometimes 1,000 atoms. So that happened in the span of 30 years. So what do we do? So if you think about the the computation. So think about an enzyme. An enzyme has about an average of 3,000 atoms. In order to compute that particular enzyme, it could take about a year to actually get a result. So what we do is we carve out a small piece of that enzyme, hopefully the part that actually makes sense for the actual model. So we call these theoretical models, and we try to simulate what happens in that particular enzyme based off of that cutout. And so the idea is to increase our efficiency, but also to reduce computational cost. We use physical concepts. For those who are in the room who are physicists, I'm sorry, quantum mechanics, all of that. So we use physical concepts to try to understand and solve our molecules, but for the most part, we're really trying to, in many ways, just model like a small portion of like that particular big enzyme and try to understand, see if we can actually get those answers without going through the whole computational course. I'm not sure if that's what you asked me.
Yeah, that's great.
Okay, great. It just went up for now.
So how do you know if you're right?
Okay, it's like my dissertation. All right. So this is actually a question that dogs theoreticians and in regards to the field. And so I was actually at a dinner party with my partner it's mostly filled with English faculty members, and I got asked the exact same question: how do you know if you're right? And I said something that I'm not sure. I need to like probe and to make sure that this actually makes sense. But the way I thought about it is that as a theoretician, I'm very invested in telling you what's true, at least mathematically. But I can't tell you what's real in the sense that at no point in my process have I actually observed a molecule do what it does in real life. The only way I can tell you what's real is if an experimentalist goes into the lab and actually mixes those things up and supports the model that I bring. And so a lot of my lab is very collaborative. All the projects I do usually have experimentalists as well who are working on either reproducing our models or supporting or refuting the models. And usually it's a back and forth. We come up with a whole model for how this reaction will work. The experimentalist goes into the lab and then actually tests it out. If it works, they figure out a whole new concept that we can then bring back and feed back into our model. And so a lot of this is very cyclical across computations and experiments. We can get into philosophical discussions about truth and reality, but I think in general, theoreticians are really focused on what can be mathematically said as true, but unless you can observe it, it remains an imagination, right?
As an experimentalist, yes, that's absolutely right. So we'll do probably one more question, and then I think we'll take a break. Is that about right, Sarah? How do you train students to think that way? So what you're doing is you're taking your skill sets in computation and you're developing collaborations with experimentalists, which means that you need to understand how those experimentalists think and understand the sort of data they generate. How are you engaging students in that fairly complicated and nuanced thought process?
Well, it goes in several ways. I think, first of all, I'm a little bit spoiled because I have incredibly brilliant students in my lab. But structurally, the way this happens, that I've been infusing computational chemistry from the very first year students get into Chapman. And so one of the things that we're doing in our department is having all of our students take a computational-based lab every single year, from their general to the organic to the physical chemistry, so basically year 1, 2, and 3. And so the goal is that they continuously see how theory can either influence the way they see experiments, or it can help validate their hypotheses from what they're observing. And so there's a lot of that being fused in pedagogically. In our lab, I think you're right. It's a whole new thought, not just thinking about your own research, but thinking about how that influences another person's research lab. So a lot of, I think the troopers over here, they consistently present their work, and they consistently present it to people who are not in the lab, people who are not theoreticians, because I think one thing we could do as scientists, we can kind of stay in our own heads. And then, when we actually have to sell our work or try to tell people what we're doing, it gets really difficult. And so a lot of what we're doing in our lab is to practice that every single day. And so I want to shout out Allegra, who's a synthetic organic chemist as well, who came in with me. And one things we're planning on doing in the summer is to actually have these super group meetings where our computational students are presented to the experimental students and vice versa, because a lot of that is actually getting them to see how their work actually applies across different fields. So.
Perfect. So by next fall, we should have Chapman chemists who are both true and real with their science, which I think is a bold step forward for us. I think what we'll do is we'll take a break, give people a chance to refill their beers. Remember, write some questions on those cards. We'll come through and collect those where people are refilling their glasses, and then we'll come back and answer some of those questions in the second part tonight. Thank you so much. Right, so I think we have more full glasses than empty glasses, which means that we're about ready to start. Thanks for sticking around, everybody. So one of the things that we didn't get to in the first half was a really important question. Maduka is a new faculty member, and as such knows that he needs to play the politics of being a new faculty member. He noticed Helen Norris here tonight. And he said, I need to make sure that I talk about the computational infrastructure that Helen has provided for my success. And so I wanted to start off with that question. Can you tell us a little bit about the computational infrastructure that Helen Norris has provided for your success?
You blew my cover, Jason. No, but I actually did want to talk about that because I think it's... without the infrastructure, you can't do research, right? Without the actual equipment. And so one thing I've been really, really lucky to have Chapman and Schmidt College actually built a whole high-performance computing cluster for me and maybe other researchers. And I've been really grateful about that because with that, it's basically about 576 computing cores, which is like, think about 576 computers all connected. And then so you can run your jobs in parallel and you can do computations that you would never think about doing, right? Take a big enzyme or take a small enzyme or what have you and split that job into 26, 36 computers so they all run in parallel. So something that would take, months to get data from, it takes maybe 8 hours. And so that really dramatically speeds up the productivity of my research. So I did definitely want to mention that, even though in a first year faculty, dutiful, thankful way, but I'm really grateful for that experience.
It was much more smooth when he told me to ask it subtly, but so okay. Okay, so a real question. When you run your computational models, what types of output do you actually receive, and what do those output represent? Who asked that one? All right, beer back here.
Nice. No, that's actually a fantastic question. So maybe I can start with the input. It's a text file.
Wow, a text file.
I know, so fancy, right? It's absolutely a text file with just numbers and words. And then the computer and the program parses that text file, does a bunch of computations, and what you get back is, surprise, surprise, another text file. But in this text file is really where all of the I keep using the word magic, and I'm a scientist, I shouldn't use that word all the time. But that's really where everything happens. You can essentially take that file, and then programs could transform that file to a three-dimensional molecule. And then you can view the molecule, rotate it around, figure out all the thermodynamics, that's a word, but figure out the energies around that molecule, how that molecule, quote unquote, moves. You can hypothesize about what kinds of interactions in that molecule are keeping it the way it is by actually visualizing it. A lot of our lab, as my students will probably attest to, is also dealing with spreadsheets. So you're dealing with energies all the time, and you're trying to compute and analyze the energetic quantities in your molecule. So a lot of it is about text and numbers, but then the fun part is when you actually get to visualize the molecule and actually see in many ways what is going on in between reactant and product, or first and second.
All right, so next question. Can you give us some thoughts into recent successfully synthetic enzymes created? Are there any that are particularly mind-blowing? And are there any that are on the near horizon that you're aware of? Whose question is that? Right here.
Okay, that's a big question.
Just the mind-blowing ones.
That's right.
Focus
That's
on that.
right. Well, I think there's a lot of work on synthetic enzymes, and that's not necessarily my field, but I think the ones that fascinate me are the enzymes that do things that the body or the natural state of that enzyme will not do. So I'm talking about emulating, right? So I'm studying how enzymes work so I can emulate what they do. But there's some enzymes that have been designed that do things that are more powerful or different than actually what it's naturally supposed to be doing. So there's a reaction called the Diels-Alder, don't worry about it. But it's called Diels-Alder. It's a synthetic reaction that the folks, my academic, we call it academic parent, but my academic grandparents actually worked with another person, I think is the next question in this pack, worked with another scientist in the University of Washington to design and create this enzyme that does a reaction that the body would, or the organism would not naturally do. And that, to me, that's more powerful. Because that means you're not just learning from nature, you are now also influencing it with your human mind. And so that's about all I can tell you about that state.
Awesome. Okay. I may have mixed up the order of these. So if you were thinking that you knew the next one, you may or may not know the next. That's right.
It's
All right.
fun.
All right, here we go. Okay. So as a computational organic chemist, what is your opinion on crowdsourcing your research? For example, like Fold It. This game uses people's combined brain power to find the optimal folding sequence.
It's like
Wait, whose is that? Your initials are AE.
great. Yeah.
Right over here.
It's like Jedi, because I did know that was the next point. So there is a faculty member and a scientist in University of Washington. His name is David Baker, and he's the one who created this program called Fold It. And essentially what this is you can think about it as a game. So you take what is essentially one of the holy grails of biology and chemistry, which is to predict function of enzymes from just looking at the structure, and he crowdsourced it. He had gamers who are in the gaming industry essentially play this game where they try to figure out the folding patterns of proteins. And so it was interesting. There was this one particular protein, I can't remember what it's called, but nobody could figure out what that protein actually looked like. And he outsourced it to a whole bunch, completely crowdsourced, and they figured out the actual folding pattern of that protein. That paper, I think, has upwards of 35 to 40 authors on that paper because of all the people who actually had a hand in from discovering the actual structure of that protein. So I think it could be very powerful. I think the idea of opening up your science and having people who may not be in that field participate and use their own skill sets could be very powerful. It's hard. I don't have the expertise to know how to do that, but that's something I'm certainly willing to think about and encourage in my own lab and my own experience.
I'm going to ask a follow-up on that. Is that an investment in the crowdsourcing? Is that people's brain power, or is it computational power, or is it both?
Right, both. So there are, so that's an example of people's brain power, right? Just bringing in people who are not scientists to do scientific work. But there's also folks who are spreading out. So there are these crowdsourcing machines where you can just register your own computer to a database and have your computer be used for research. And so I don't remember exactly who is doing this. It's run off the top of my head, but there are foundations built on this idea that anyone can just donate their computer to the cloud and have computations be run on them. So that's another way to crowdsource not just the brain, but also crowdsource the equipment.
So anybody here could be number 58 in the cluster tonight? That's right, number 50. Oh.
My students.
All right, so next question here. What's your favorite part of the research that you do, and how has Chapman impacted that part of your research? And who asked this question? Yeah, I'm just teasing. I'm totally giving a free drink to the students. Hey, soda.
this is gonna get sappy, but my favorite part of my research, and I'm not saying it because they're here, but so it was, I just started as a faculty member, and I think one of the biggest joys I had was last Friday when my students gave their second group presentations. And it's so, like, seems so minimal to see that. But so somebody who was just a student, who has gone through this gruesome process of being a faculty member, and then turning right around and seeing your students not only learn, but impact knowledge on you as a faculty member. That, like, yeah, I was definitely about to tear up in front of them, but I didn't. So, but it is that, I think that's probably the best part of research here at Chapman, is that it's still focused on students learning, even if you are doing quote unquote good work and publishing and all of that. A lot of it is, a lot of us are still thinking about, like, how is this actually impacting our students? Like, how are they learning from this experience? So to me, that's my favorite part.
That seems like a fine place to end this conversation to me. Let me thank Maduka for being here tonight. Once again, thanks everybody for coming out tonight. Thanks to Chapman Crafted for hosting. Thanks for Panther Production for making us look and sound good, to use the Andrew Lyon quote. And thanks for all of the Schmid staff, Elisa, Sarah, parent volunteers, for making us have such a nice event tonight. And Bonnie for chips, a second time. We have two more excellent speakers coming up in this season's Science on Tap. On Monday, the 11th of March, we have Dr. Doug Fudge, who is here tonight. Yes? Doug's going to be talking about hagfish biomimetics. We will unpack that early on the 11th, don't worry. Hagfish biomimetics, profound lessons from the deep sea. So another talk focused on lessons from nature. Do people know what hagfish are? There's these fish that create just gallons of slime when you touch them or when a shark bites them. And yeah, they're super cool. It's going to be a really good one. And then on the 15th of April, we have Mackenzie Krieger, who is going to be talking about Your Sustainable Home. Mackenzie is actually the Sustainability and Energy Coordinator on campus, and she'll be sharing a little bit about how we think about that on campus and with the Environmental Science Policy Program. So we hope to see all of you back at both of those events. Again, thank you for being here tonight. And one more time, thanks so much, Maduka, for being with us tonight.